Hello my dear student, hope you are fine and safe at your home. Today I am going to take a review class about cell division. Hope you will enjoy this class. After watching this video, if you have any question about cell division, please write down on comments box. I will try to give your answer as soon as possible. Welcome again. The cell is the structural and functional unit of life. New cells arise from the pre-existing ones. The process by which new cells are formed from the pre-existing cells is called cell division. There are two types of cell division, mitosis and meiosis. Amitosis also a type of cell division, but according to the modern concept, it is called binary cell division. Today I am going to describe about mitosis and meiosis cell division. Mitosis cell division is called equational cell division, dividing the mother cell into two daughter cells which are identical to each other and also to the original mother cell in every respect. The first stage of mitosis cell division is prophase process. Prophase is an early process. The chromatin network becomes visible as it condenses into separate threads or chromosomes. At this stage, each chromosome appears as a very fine long single thread, the chromonema, and is it described as the monad. The nucleus envelope and nuclear are prominently visible. At this stage, each chromosome is shorter, thicker, and consists of two identical sister chromatids joined together by a spherical body called a centromere. Second stage is prometaphase. Prometaphase is the second phase of mitosis, the process that separates the duplicate genetic materials carried in the nucleus of a parent cell into two identical daughter cells. The breakdown of the nuclear involves frees the sister chromatids from the nucleus, which is necessary for the separating the nuclear materials into two cells. Other spindle fiber elongated, but instead of attaching to chromosomes, overlaps each other at the cell center. Prometaphase is followed by the third phase of mitosis known as metaphase. The chromosomes are moved and get arranged in a plane along the equatorial of the spindle. This result is the formation of equatorial plate. The centromere of each chromosomes in the plate is connected with both the poles by the kinetophore fiber. And next, anaphase. The centromere of each chromosome divided longitudinally into two. During late anaphase, the two groups of daughter chromosomes are pulled away from each other and starts moving towards the opposite poles. Most probably, the movement is caused by the shortening of the chromosomal fiber. In each group, chromosome appears V, L, I, and J-shaped as the centromeres are pulled towards the poles and the chromosomal arms tail behind. The last phase is named telophase. The two sets of daughter chromosomes reach the opposite poles. The chromosomes again become long and thin. The spindle fiber also dissolved and disappeared gradually. The two daughter nuclei are identical in structure and characters. They are also exact copies of the original parent's nucleus. And finally, cytokinesis is the division of the cytoplasm into two separate cells. It is usually occurs in telophase along with the formation of daughter nuclei after the nuclear division. Cytokinesis is accomplished by the formation of phragmoplast and cell plate at the equatorial of the dividing cell. And now I am going to describe about meiosis cell division. In sexual reproduction, organisms, two important phenomena regulate the number of chromosomes in their life cycle. There are meiosis and fertilization. Meiosis is the reduction division in which the diploid number of chromosome is reduced to haploid during gamete formation, whereas in fertilization, two haploid gametes fuse to form a diploid zygote. In this way, the diploid condition is restored again in the life cycle. So we can say meiosis is a spherical type of division characteristics of reproductive cells in which the diploid number of chromosome is reduced to haploid in the daughter cell. In the meiosis, chromosomes divided once, while the nuclei, or you can say nucleus, divides twice. 4 haploid daughter cell results from one diploid mother cell. These differ from each other as well as form the mother cell. So in a higher plant, meiosis occurs in diploid mother cell, and lower plants it occurs into diploid zygote. The meiosis cell division is a continuous process. For convenience of description, it is divided into two main types, meiosis 1 and meiosis 2. So the various events in meiosis 1 are classified into like prophase 1, metaphase 1, anaphase 1 and telophase 1. So in a higher class, you will get more idea about meiosis cell division. So meiosis cell division, the karyokinesis consists of two complete nuclear division called the first meiotic division, meiosis 1, and second meiosis division is called meiosis 2. Especially meiosis 1 is reductional division involving separation of homologous chromosome. So that's all about today's lecture. Hope you have understood about cell division. If you have any question, please write down on comment box. I will try to give your answer as soon as possible. Good day to all.